I love turtles, so.
Thanks very much.
All right, I'm just going to give it a minute here in case somebody's waiting for this one and they want to hop on. So our next speaker is Stuart Fetherson. Stuart is a graduate research assistant in the Department of Natural Resource Management at SDSU. He's studying the population genetics of bobcats in South Dakota. His professional interests include management of critical habitat, conservation biology, and landscape genetics. And the title of his presentation today is Research in Progress, Population Genetic Structure of Bobcats in South Dakota using harvested samples to inform management. And students, Stuart, you can go ahead.
Okay, thank you very much. So let's see. Can you see this all right?
Yes.
Okay, perfect. So yeah, my name is Stuart Featherstone. I'm a second year master's student at South Dakota State University, and I'll be discussing some of the results I have from my first chapter, talking about population genetic structure of bobcats in the state using harvested samples. And so for a little bit of background information, bobcats are one of two species in the genus Lynx in North America. They're the most widely distributed native felid in North America. They're mesocarnivores, and on average, they weigh about 6.5 to 10 kilograms. In addition to that, they're hypercarnivorous. Most of their diet is made up of meat. And in general, their habitat consists of areas with abundant cover that facilitates their stalking and ambush style hunting. So historically, bobcats have ranged throughout the contiguous United States as well as areas of Canada and Mexico. And currently, they maintain about 80% of that historical range. So bobcats are listed as a species of least concern on the IUCN Red List, and throughout their range, their populations are stable or increasing. Despite this, they're also listed as a CITES Appendix 2 species, which is reserved for those species that could potentially be threatened if trade is unregulated, as well as lookalike species. Because bobcats are the only species of spotted cat in the world that can be legally traded, having stronger protections on them also provides stronger protections for other threatened and endangered cats. And so in South Dakota, harvest of bobcats has been managed since 1975. And in the period from 75 to 77, the entire state was open to harvest. And then from 1977 till 2012, only those counties that are west of the Missouri River were open to harvest. And from 2012 to 2020, a number of counties east of the Missouri River were open for harvest, albeit with a shorter season and a bag limit of 1. And currently the entire state is open for harvest again, but with the same lowered bag limit. And so we can look broadly at the genetic structure of bobcats based on the mitochondrial DNA. Specifically, Dawn Redding did some work where she found 2 distinct clades of bobcats, an east and a west. And likely this differentiation was caused by climactic events during the mid-Pleistocene, during glaciation at that time, that created an eastern and western refugia for the species. And currently we're seeing through the Great Plains a sort of suture zone where these two formerly isolated populations are interacting once again. And so if we look at the nuclear DNA, we can get an even finer scale picture of structure. Specifically in the Midwest, Redding identified 6 distinct populations, genetic populations of bobcats. And notable here is South Dakota was the only state in the Midwest that fell into that western clade from the mitochondrial DNA. And this is also important to note that she didn't have any samples from east of the Missouri River either. And so the goals of my project are to inform management units by identifying discrete bobcat populations within the state. And more specifically, I want to identify the patterns of this population genetic structure and compare these findings to similar research on bobcat population genetics. And so all of my data comes from tissue that was sampled from harvested bobcats between 2014 and 2019. All samples were collected by South Dakota Game Fish and Parks when harvested bobcats were taken in to receive an export tag in accordance with the CITES regulations. All of my laboratory work was done at the University of Idaho's Laboratory for Ecological Evolutionary and Conservation Genetics, and DNA was extracted from all samples by staff there prior to my arrival. Once I was there working on the project, I selected 17 microsatellite loci that would be suitable for the analysis of this project. And then each sample was amplified at each of those 17 microsatellite loci using polymerase chain reaction, or PCR. And then finally, I called alleles for each sample using the program Gene Mapper and created genotypes for each sample. So once I had that genetic data, I had organized the consensus genotypes using package ConGenR, and then also quantified a number of diversity metrics such as heterozygosity using the package GenePop, both of which in program R. And I used the program Genelex to quantify a probability of identity, specifically probability of identity for siblings. Because these are harvested samples, I obviously knew that each sample represented an individual, but this would give me a threshold for the minimum number of loci a sample would have to contain a genotype at to be informative going forward. And specifically, the identification of siblings is a more conservative estimate and accounts for relatedness in the population. And so closely related individuals in a sample can bias some genetic structuring programs. And so I also calculated relatedness between all individuals in the sample. So to evaluate genetic structure, I used two competing approaches, both of which are Bayesian clustering algorithms. The first of which is program structure, and the second is the Bayesian analysis of population structure, or BAPS. And then along with these, I used two measures to quantify differentiation between population clusters. And for those, I used FST and its derivative GST prime. So I had to finalize the sample that I was going to use for analysis. I had 1,225 samples that had successfully amplified at any number of loci. From there, using the probability of identification for siblings and relatedness, I removed individuals that did not meet our criteria down to 878 samples. And then finally, I removed any samples that did not have location information. And in this case, it was the county of harvest. So next, I want to look for evidence of genetic structure in the state, which there was. Our rates of observed heterozygosity were less than expected. There were instances of linkage disequilibrium, and 12 of our 17 loci were out of Hardy-Weinberg equilibrium. And all of that's just to say those could be a result of the Walland effect, which just suggests that there's a level of structure in the population that isn't being accounted for when analyzing it all together. And so with evidence of structure, I went ahead and ran the two Bayesian clustering algorithms, the first of which being program structure. And so both of these programs represent the most likely number of distinct genetic clusters by the value K. For structure, I used the test for delta K to analyze the most likely number of clusters. And so you can see here at peak at K equal to 2, which we have to be cautious when interpreting because delta K often has a bias towards K equal to 2. And so there's also support for K equal to 4 and 10. And so support for 4 populations or 10 populations as well. And then I ran two different models in BAPS. First of all was clustering of individuals, and that one used the same information that was used in the structure run. And that also came up with K equal to 10. And then I ran a spatially explicit model, the clustering of individuals with spatial data for BAPS, which came up with a result of K equal to 2, which provides further support for structures finding of K2. Now, because the level of spatial data I had was only at the county level, to run the spatial model, I created a buffer at the center of each county, and I randomly distributed points within those buffers for coordinates for the model. And so next I wanted to see what all of those results meant and assess whether the clusters as inferred by these programs made sense geographically and biologically. And so here you can see each county is colored according to whichever cluster a majority of the individuals harvested in that county assigned to. And then more specifically, the pie charts in each county show the actual proportion of individuals in that county and where they assigned. And so these results make sense. The clusters translate well to a coherent layout on the landscape. They're not just randomly dispersed about. And so when we look at the results for BAPS K=2 as well, we see a remarkably similar pattern. And in both cases, you're seeing this east to west divide, where on the peripheries, they're much more insular. And as you go towards where they meet, you can see more individuals of each cluster in those counties. And this is interesting to note also that we see this east to west divide, but it isn't necessarily at the Missouri River. The actual boundary appears that it could be more west of that, or that the counties on the west bordering are more related to those in the western counties. And so as I mentioned, I was going to look at FST and GST prime for differentiation. What's important here to note mainly is that at a value of 0, the populations are identical. And the further you get away from 0, the more differentiated they are. And so because GST prime, it has less bias when interpreting highly variable microsatellite data, I'll be interpreting that. And so in each table going forward, the values shaded in gray will be the GST prime values. And all GST prime values that I present here were significantly different from 0 at a p-value less than or equal to 0.001. So then we can take a step back and look at what support there was for k=4. And again, that same pattern appears here, where that first cluster, those eastern counties, appear remarkably similar again. And that second cluster has now been split into 3. And also interesting to note here is the blue cluster, where the Black Hills falls out as its own distinct population. And then again, if you look at the GST prime values, the cluster in red, that eastern cluster, is more different from every other cluster than they are to one another. And so finally, things continue to break up more, and it gets a little messier at k=10, but a lot of those same patterns are still recognizable even at this level. 2 things I'd like to point out in this one are cluster number 6, the maroon, because that one's actually overrepresented on the map. It doesn't actually have enough individuals to really characterize it as a genetic cluster, but 2 of the counties, it was the majority or tied for majority. And that's compared to cluster number 10, the black cluster, where it has a sufficient number of individuals, but at no point was it the majority in any of these counties, although it's still spatially clustered very close together. It's not dispersed throughout the counties by any means. And so again, we can look, and cluster 4, the red here, the major eastern cluster that we've seen is more different from all of the other counties, as we saw at levels of K2 and K4. And now we're seeing this gray county in the center that's sort of at the convergence of all these other clusters, and you see more individuals from different clusters making up these counties. And you see the least amount of differentiation here as well. And so then we can switch over and look to the BAPS K10 results. Here, it's important to point out that, again, we have two clusters that don't have a sufficient number of individuals to really characterize them. And you can also see how this biases the GST prime values high. And so we have to take that into account when interpreting these as well. But all of the individuals in the black cluster were found up in the northwesternmost county, and all of the individuals in the white cluster were found in the section of the Black Hills that's in Fall River. And then again, we can look, and this red cluster.
Yeah, I've never seen anything like it.
Cluster number 10
Atlantic Flyway is a weird
is
place.
also the most different from all the others. And looking at 4, that major gray cluster there is the least differentiated from all others. And so the implications of this are there's large support for 2 populations, and then beyond that, evidence of even finer levels of structure, which may not necessarily be what we would expect for a wide-ranging carnivore, that we would see these levels of genetic structure in the state. And so this information could be used by managers to potentially inform management strategies going forward and populations to pay attention to. Bobcats also just provide a very unique management situation in that they are at this increasing or stable level where they can sustain harvest, but are also regulated closely enough that researchers can have access to this kind of data. And so this project really is due to the cooperation between sportsmen, researchers, and management agencies. And then again, as I mentioned earlier on, the major studies done on bobcat population genetics didn't have access to any samples east of the Missouri River. And so it's interesting to see how these results relate to those of other studies on bobcats in the Midwest. And so finally, I would like to thank everyone on my committee, Dr. Lonsinger, Dr. Jenks, Dr. Lehman, Dr. Perkins, Dr. Waits, and Dr. Hatfield, as well as everyone at the Laboratory for Ecological, Evolutionary, and Conservation Genetics, whose expertise was extremely useful throughout this entire process. And then my funding agencies, GFP and the USDA. Thank you.
Thank you, Stuart. Does anybody have questions for Stuart? We have about 3 minutes.
Oops. Okay.